So did you have any preference? I mean, of course, people who are coming to a cryonics meeting or who are members of cryonics organisations have got a natural interest in health and longevity. So I presume that means that the most interesting stuff is going to be your work on Progeria. Do you think that's correct?
It might be. I mean, I'll give you a brief summary of my interest in cryonics, how it developed and where it led me.
Yeah.
Okay.
And did you? How long have you been interested in cryonics? Is that something that you got interested in more later on, or have you always been interested in that sort of idea? Because it's been around since the early 60s, I guess.
Yeah. Well, I'll start. I was born and raised in Missoula, Montana, and when I was in high school, which I entered in 1960. Sometime around 1961 or 62, I read an article in Playboy magazine. I don't remember who wrote it. It might have been Edinger. It was on the idea of freezing and coming back in the future. And I thought that was a really interesting idea. And it got me thinking about the idea of freezing. So I did some reading on it and freezing, and I did an experiment where I think it was hamsters, could have been white rats, but I put them in the freezer because I'd read you could freeze them for 45 minutes or so, and to the point where 70% of the water in their body was frozen as ice, and then you could warm them and they'd come back to life. And so I did that on this animal or several animals, and had a heat lamp and put it on them after I brought them out. And they came back, and it was pretty amazing.
Really? Amazing.
Yeah. Yeah. So, and then I got admitted to college, to Johns Hopkins University, where I started in '64, and got in touch with some fellow, and I can't remember his name, but he was very interested in the idea of freezing, and he and his wife, or partner, we talked a lot about it, and they were building a facility, I think it was in West Virginia, and I went with them once to see it, and stayed interested in the idea. And then I decided I would major in biophysics so I could learn more about freezing. And I graduated a year early and started graduate school in a PhD program in biophysics. And
So the medicine didn't come first. You actually got into the biophysics?
Yeah. So it was, I started, and it was in the graduate program at Hopkins for 2 years, and the Vietnam War had come along, and I was on a draft list. So I'd always thought I'd go to medical school, but graduating in 3 years meant it was hard to get the forms ready to be an applicant. Anyway, I applied and I got into Harvard Medical School and went up, and since I'd already had some biochemistry and I got some free time during my first year, and I went and talked with Charlie Hopkins, who ran the blood freezing unit at Mass General Hospital. And I started a research project there with him to freeze cancer cells and see what their viability was just afterwards without any preservative or something along that lines. And we found there was a certain small fraction that would survive freezing. But then I got really thinking about it, and I thought, well, even after you come back, you'd still have to deal with aging as a problem.
Yeah, that's right.
And so I got really interested more in aging and talked to the different experts at Harvard about aging, and I started research in a lab, and they were working on DNA repair in Progeria cells, and I got really interested in Progeria.
So what year would that have been?
Well, let's see, it would have been, I think, 68, 69.
Oh, so that's well before all the sequencing stuff started happening.
Yeah, it was long before that. And so after I graduated from both medical school, and I took a year off to finish my PhD in biophysics, which was dealing with neurochemistry. I worked on the visual system in the frog brain, and because that was one of the professor's areas of interest, and I thought it was interesting to learn more about the brain. And so I finished up and with my degrees, and then got a medical residency, internship residency program in New York City. My wife at the time was a concert pianist and wanted to be in New York, so we moved to New York. And I did my 3 years of training, and then looked at the geriatric programs and didn't think they were all that interesting. So I decided genetics of aging would be interesting, and got a position as a trainee and then a faculty member at Cornell Medical College, which was in New York City, and moved there, and I got a grant to study progeria. And it's a very rare disease. There's about one new case every 4 million births, so about one new case a year in the United States. And anyway, I worked on it for a couple of years and wasn't able to, find any valid biomarker that would persistently, that I could use to study it and get to the bottom of it. And it was so rare, and it's not inherited, so each case is a new mutation, a new dominant mutation. And we didn't have the technology to sequence at that time, but it was just becoming available. So I went to work I went and visited with a professor at Rockefeller University, which was next door, and started a project to move to work on Down syndrome as a model of aging. And Down syndrome has a number of features of premature aging, like progeria. And we worked on trying to clone the 25th chromosome.
Can I just ask a question there? I mean, Down syndrome is an extra chromosome, though. Isn't that going to be quite different from a single point mutation?
Definitely. Yeah, and it is, it's still, they do, Down syndrome individuals have a number of physical features that look like premature aging, and they also develop Alzheimer's disease with, at early ages, around 50 on average. And so it was, it was within the area of aging anyway, and we had access to Down syndrome cells and study. And then the head of the department was leaving, and the genetics unit at Cornell Medical School was kind of dissolving. So I looked at going to Rockefeller, and I got an appointment to start there. But then I got recruited. There was an opening at the New York State Institute for Basic Research in Developmental Disabilities, which is a state-funded research center on Staten Island. And the director there was very interested in Alzheimer's disease, and he liked my interest in progeria. So I moved there and became head of the Department of Human Genetics, where there were 5 laboratories, and I started work on Down syndrome and continued to have an interest in progeria, and had kind of set up a registry. And then I met a family that had a child with progeria, and both of the parents were doctors, and their child was 2 years old, and the wife was getting an MD PhD, and she was in her pediatric residency and decided to drop out and do research on progeria. And the family and her sister, the 2 ladies, started a family sponsored the Progeria Research Foundation to raise funds to support research into progeria, because there's very little going on. And that led to a collaboration with Francis Collins, who's now the head of the NIH. And he'd seen a patient when he was in his residency with what he thought was progeria, and turned out it wasn't, but it was something related, perhaps. Anyway, the consortium, I'd seen a patient where, actually a twin that had a chromosome rearrangement and insertion deletion of chromosome 1. So this gave us a clue that we might find a gene in this region if we could clone the breakpoints. And so we were setting out to do that. And anyway, it led to us finding the gene, the mutation that causes progeria. And this was published in, I think, well, I can't remember exactly. It was 2001 or so.
Right.
And
okay.
it was, it was quite interesting. So I'd maintained an interest in progeria, but I was, around the time that I started work at this institute, we were just learning about a new syndrome called the Fragile X syndrome, which turns out to be the most common inherited cause of mental retardation or intellectual disability. It's X-linked and runs in families. And so we started work on that, and that became the main thing that I focused on during my time at the Institute. And I was there for 35, 40 years working on Fragile X primarily, but with an interest in progeria. And I continued to maintain contact with this Progeria Research Foundation, and I'm still on their advisory board and help with reviewing grants to study progeria. And it turns out that, it's a model of aging, a human genetic model that looks like premature aging. And it's due to this single point mutation that causes an abnormal splicing event, and it leads to a buildup of an abnormal protein that's called progerin. And they have massive amounts of it, and it causes the cells to prematurely age. And we all have a little bit of progerin because of abnormal splicing, and it builds up with aging in certain tissues. So it continues to be, related to aging and of interest. But I've continued to be very interested in aging, and I retired from my position there at the Institute. I became the director after I was there for 40 years. In the last 15 years, I was the director of the Institute, and we were focused on autism and Fragile X, and I still have the interest in progeria. And I moved to Australia and got remarried to a lady who's a pediatrician here, and I became, an Australian partner. And I have been associated with the group at the University of Sydney that's studying aging, metabolism and aging at the Charles Perkins Center, and it's been quite interesting. And David Lecoutier, who's one of the co-directors there, is quite interested in aging. He's an editor of the Journal of Gerontology, which is one of the better journals. Anyway, I did a 23andMe gene screening test of myself 10 years ago or so, and found I was a carrier for the gene called ApoE4. And ApoE4 is the major genetic risk factor for Alzheimer's disease.
Yeah, I've got the same one, actually.
Yeah, about 1 out of 4 people of us European descendants are carriers. And so it gives us, a threefold increased risk of developing it. So I've been studying all about the genetics of aging and ApoE4 and the polygenetic risk scores that go along with it now, because there are some 40 other genes that modify its risk. And still am, very interested in the topic of aging and seeing if it could be reversed. And I was reading, Greg Fahey, who's been a cryobiologist, has also been interested in aging and published an article where he was giving human growth hormone to 9 subjects, 9 men, middle-aged, and along with a couple other medications, metformin and DHEA, and showing that their epigenetic clock was reversed by 2.5 years after taking it for 6 months. And so it's kind of an interesting topic that maybe you could reverse aging with something like growth hormone, or there's also some parabiosis-related experiments where they thin the blood. The convoys in California have published methods of, at least in rodents, where they can thin reduce the plasma content by 50%, leaving the albumin there, and the mice they slow down their aging process or kind of reverse the epigenetic aging. So it's still quite interesting. But I read in June, at the end of June, there was this article in the New York Times about cryonics. And it got me thinking more about it again and reading up on what's been going on. And I realized, I hadn't thought very much about it since I left medical school and was focused more on aging. But now I'm kind of hoping to learn more about what's going on and thinking more about what I should do in that area or could do.
Yeah. So are you going to be permanently resident in Australia now, are you?
Yeah. I mean, my wife's still working. She's younger than I am and isn't ready to retire. And so I'm a permanent resident living here, but I have a house that I still own on Staten Island. It's quite a grand old historic house, and I have my 2 adult children that live back in the East Coast. And so I go back. I was going back 4 or 5 times a year to take care of the house and see my kids, but couldn't go back because of the pandemic since a year ago March. Was February. A year ago February was the last time, or January was the last time I made it back. So I'm a resident here, and I don't expect, but maybe when she retires, we'll spend part-time here and part-time there.
Right, okay. Yeah, I was going to say, if you're going to be a permanent resident in Australia for the duration, then you should talk to Peter, who's online here, because he's the key person that's been setting up the Holbrook facility, and he's also in Sydney. I mean, I moved out of Sydney some years ago, and I'm in this little country town now, but before the pandemic and whatever, I was making regular trips back to Sydney, and I'd really like to catch up and talk about a dozen different things that you've been talking about, actually. A couple of questions on the genetic stuff. So when the Progeria stuff, from the early days when the sequencing technology wasn't around, and then towards the year 2000 when the Human Genome Project and other things were making progress, I presume that gave a bit of a kickstart to the Progeria research. But it sounds like you've got it fairly sorted out about what causes the problem. There's this increase in the progerin protein. But does that mean, and talking about the other stuff that you mentioned in terms of reversing aging, if everyone's got a bit of progerin floating around in their system, if you can eliminate that as well as the other things with the metformin and the human growth hormone and the other stuff, it doesn't sound like anyone's been talking about trying to get rid of the progerin yet. It's these other things so far.
Yeah. Well, it's historically, what we had a conference on progeria of anybody that was interested in it, and Francis Collins came, and he had a graduate student or a postdoc that was working with him who got interested in it, and they decided they'd look for homozygosity in the cell lines that were available. I stored about 10 different patients' cell lines, and they got a collection of 18 cell lines, and we're going to do homozygosity mapping. And I thought that was unlikely because I was convinced it was a dominant new mutation.
Yeah, that's right. Yeah.
But they went ahead with it, and at the time, there was another disease called mandibular acral dysplasia, or MAD syndrome. This kind of gets confused with progeria because they have some of the same features, but they're quite different. And it was found, it's recessive, and the gene for that was found to be lamin AC in a Sicilian family study that was done. And so that was right in the area where we had been thinking the gene might be located because of the rearrangement of the chromosome in the patient I'd had access to. And so he got very busy with the sequencing facilities that they had at NIH, was able to sequence the lamin gene in the 18 patients, the subjects that they had the cell lines on, and found a common mutation that was present. And it was a single nucleotide change that caused a splicing event. And it spliced out about 50 amino acids of the mature lamin A protein, and that leads to a retention of a lipid group on the end of the protein, a farnesyl group that stays around, and that gums up the works and makes it an abnormal, and causes blebs of the nuclear membrane. It's a nuclear membrane protein and very important. But so looking at it in the so we know quite a bit about how it's caused and there are a lot of interesting new treatments that are being developed for it. It doesn't seem to be present in very high quantities in human tissues as we age, except in some endothelial tissues and in maybe fat cells. And so I did a study here on the group at the University of Sydney have been pioneering and seeing what's better for aging in mice. And they had found that a low protein diet, can double their lifespan or increase it by 50% anyway. And so they had mice that were, on a special diet that I thought, well, let's see what the progerin levels are in normal mice and in these mice that are on the diet that promotes longevity. And but we really found that there was very, very low levels and not enough that we could measure adequately in the tissues that we had available in the mice. And so we kind of have given up. I think that there are methods that could be looked at, but I don't think it's turning out to be likely to be a very significant factor with aging. That's why there hasn't been much done of it in terms of, human aging.
Yeah, well, I suppose there's a number of other things that are going to be a bigger factor to begin with, just for normal, healthy people, not people that have got some sort of genetic illness. Tell me, do you know about a thing called Batten's disease?
Yeah.
Yeah. I've got that gene in the family as well on my father's side.
Yeah.
I had a cousin's son die of it a couple of decades ago. I know there's a few varieties of that, but it sounded similar to the sort of thing you were describing.
Yeah. It's
a metabolic thing.
It's Batten's disease. There are about 7 or 10 different genes that have been identified that cause. Yeah,
and they're causing different
progressions. Yeah. Something like Batten's disease. Batten's is the most common of the lipofuscinoses, I think.
Yeah.
Sure. I'm very interested in cryonics and in the field of aging and thinking about it. And I have, no qualms about, my identifying my interest. And, it's really what got me into science and into medicine. I was going to be a doctor to do freezing. That was my goal when I was in college.
Exactly the right thing. Yeah.